The overall goal of this procedure is to capture the formation of LC3-positive autophagosomes in conjunction with omegasomes and lysosomes. This is accomplished by using HEC293T cells that stably express GFP-DFCP1, a marker for omegasomes, and transfecting them with CFP-LC31 and marking the lysosomes with red lysotracker. The second step is to prepare the incubation chamber. Next, the incubation chamber is placed on the microscope stage, and the rich medium is replaced by starvation medium to induce the autophagy response. The final step is to select the appropriate cells and set the parameters of the video microscopy. Ultimately, live cell imaging is used to show that the LC3-positive autophagosomes originate from the DFCP1-positive omegasomes, and they eventually fuse with the lysosomes.
The main advantage of this technique over existing methods like fixed cell imaging is that we can carefully monitor the progression of events in the same cells over time.
This method can help answer key questions in the autophagy field, such as the course of events leading to autophagosome formation.
This study utilizes a HEC293T cell line that stably expresses GFP-DFCP1, a marker for omegasomes, which are precursor structures leading to autophagosome formation. 48 hours prior to live cell imaging, see the low passage number of the cells on 22 mm round cover slips in DMEM. Culture overnight at 37 degrees Celsius in 5% carbon dioxide to a confluency of 30 to 40%. On the following day, prepare the transfection complex mix for each plate, containing Optimum-1 reduced serum medium, extreme G9 DNA transfection reagent, and PECFP LC3 plasmid DNA. LC3